This lesson's about incomplete dominance, codominance, and the ABO blood groups. Now, in previous lessons, we've talked about dominance and recessiveness, and we've said that you can tell which allele is dominant by looking at the heterozygote. The heterozygote has the phenotype of the dominant allele. What we were talking about there was complete dominance, and so we need to talk about two more variations, incomplete and codominance. First, let's talk about incomplete dominance. In incomplete dominance, the phenotype of the heterozygote is intermediate between the phenotypes of the two homozygotes. Let me give you an example. Imagine that you had a single gene that controlled color in the petals of flowers, so that a red flower was homozygous dominant and a white flower was homozygous recessive. Let's say that the genotype of this is big R, and this is little r, little r. If we were to cross those, we'll call this the P generation, then the F1 generation, because it would be a generation of heterozygotes, would have a phenotype in between red and white. Heterozygotes would be pink. And now if we were to take two of the pink heterozygotes from the F1 generation and cross them so that our Punnett square would look like this, we would get homozygous dominant, heterozygous, and homozygous recessive. And our homozygous dominant would be red again. Our homozygous recessive, we would see white again in the F2 generation. This is our F2 generation inside the Punnett square. And the two heterozygotes would again be pink. So in the F2 generation, we wouldn't have the 3-to-1 ratio of phenotypes that we saw in the complete dominance setup. We would see a ratio of 1 red to 2 pink to 1 white. Previously, with complete dominance, these two had been lumped together because the phenotype of the heterozygote was the same as the phenotype of the homozygous dominant, but that's not the case in incomplete dominance. Now, just a mnemonic, I remember incomplete dominance as being intermediate or in between. In contrast, in codominance, the phenotype of the heterozygote is a combination of both of those of the homozygotes. So what does that mean? That means that in this case, the heterozygote, RR, would be a combination of red and white. For example, it might be red and white striped. And if we took two heterozygotes and we crossed them, we'd see a 1 to 2-to-1 ratio of 1 red to 2 red and white striped to 1 white. So that's codominance. Again, with codominance, you get a combination of the phenotypes of the homozygotes. Now, ABO blood groups in humans are an example of codominance, and they're also an example of multiple alleles, multiple different alleles at one locus and controlling one trait. So the alleles are IA, which we write like this with a capital I and an A, IB, and lowercase I. And these two are written with capital letters because they are codominant with each other, but they are dominant over the lowercase I, which is the recessive allele. And the alleles code for carbohydrates on the red blood cell membrane. And more specifically, really, they code for enzymes that put those carbohydrates on there. The A allele codes for the A-type carbohydrate, the B codes for the B-type carbohydrate, and the lowercase I doesn't code for any carbohydrate at all. If we now combine those alleles into all the possible combinations, we could have two A alleles, or an A allele with the recessive lowercase I. In either case here, we're going to get the A-type carbohydrate put on the red blood cells. Remember, A is dominant to the recessive lowercase I. And we would call this blood type A. Just a note that the carbohydrates on the red blood cells can stimulate the production of antibodies, an immune response, and anything that can stimulate the production of antibodies is called an antigen. So you'll hear this called the A antigen. The B allele could be present in the homozygous form, or it could also be present with the recessive I allele. That would be blood type B because you have the B-type antigen, the B-type carbohydrate on the red blood cells. You could have one of each the A and the B allele, and that's blood type AB, because remember, these are codominant. You get the expression of the phenotype associated with both of these alleles on the heterozygote. So this is our heterozygote, and this individual would have A-type antigens, and they would also have B-type antigens on their red blood cells. And finally, you could have the two recessive alleles. This individual is blood type O, and they have no carbohydrates on their red blood cells. Now, humans will develop antibodies which help trigger an immune response to any antigens that they themselves don't have on their red blood cells. Meaning, if you have A antigens but no B antigens, you will form anti-B antibodies and have those circulating in your blood. That would cause you to trigger an immune response if you were given B-type blood with the B antigen. So a person who has type A blood has anti-B antibodies circulating in their blood and can't receive blood from a type B person because that would trigger a kind of allergic reaction, a transfusion reaction. Similarly, someone with only B-type antigens will have anti-A antibodies because they don't have any A antigens on their own red blood cells. Someone who has both A and B antigens, however, will have no antibodies to either variety because they have learned not to react to their own cells, and therefore they don't have any anti-A or any anti-B antibodies. However, the person with no antigens on their own red blood cells will end up having both anti-A and anti-B antibodies because they don't have either of these antigens themselves. And this governs who can give blood to whom. You might have heard that people with O-type blood are called universal donors because when you're giving a transfusion, the recipient must not have any serum antibodies to the donor antigens, to the donor carbohydrates. And so someone with O-type blood has no antigens at all. And so there's no risk of the recipient having an antibody to antigens on red blood cells they're receiving from an O-type person. In contrast, however, someone with type O blood has lots of antibodies circulating, both anti-A and anti-B antibodies, and so they could only get blood from another type O person. If they were to get blood from a person with type B blood, for example, that blood has B antigens, B carbohydrates on the red blood cells. They have anti-B antibodies, and they would have a transfusion reaction. So that's an introduction to incomplete dominance, codominance, and the ABO blood group system in humans.